So hello and welcome everyone. We're so excited to have you all here to join us for our May Neuro and webinar, Neuro and Engineering. We're so excited to introduce our guest speaker for this month's webinar, Dr. Alex Kwan.
Dr. Kwan is an associate professor at the Meinig School of Biomedical Engineering at Cornell University, where he studies neurobiology of psychiatric drugs. His research combines optical imaging, behavioral analysis, and computational tools to understand how compounds like ketamine and psychedelics alter brain circuits to treat depression. We're so fortunate to be joined by such an incredible speaker here today, and I'm going to hand it off to Dr. Kwan here to tell us a little bit more about his career journey and experiences.
Yeah, thank you for that introduction, Sophia, and good to meet you here as well, Irene. But let me try to share my screen to start. Okay, I think you should be able to hear me and see the screen.
Yep, we're able to see your screen.
Good. Um, yeah, so I will start by saying I don't actually have that many slides. So I have about 35 slides or so. So hopefully, I usually go about one slide per minute, so maybe 35 minutes. If you have any question, definitely leave in a chat. We can happy to talk about it at the end, or yeah, definitely ask a lot of questions. Yeah, I would much rather this to be more interactive than mostly just me talking. The talk today, the presentation and the slide are divided into three parts. As I already briefly mentioned, first I'll talk a bit about what does it mean to be a neuroengineer, or what is the discipline of neuroengineering, because it's still a really nascent field. It's a very young field. I have to say, when I was in high school, I don't feel that it exists. I don't think there is such thing as neuroengineering. So I think it's good to talk about what it is and where we think it's going. Then I'll tell you a bit about what my lab does. So what specific research does my lab do and how does that fit into the general scheme of neuroengineering? And then finally, I'll tell you a bit about how I got here, which might, give you some idea, although it's, one person's experience, but at least it gives you some, thinking on how that happened. In terms of neuroengineering, I'll start by posting a question. This is Zoom, so it's hard to get engagement, but I'll ask, what does a neuroengineering do? And in my mind, if you think about what a neuroengineer or brain engineer do, sometimes I go to movies, and if you think about what movies tend to portray, there's one very common example. I don't know how many of you have watched this. This is an older movie now, Eternal Sunshine of the Spotless Mind with Jim Carrey, where Jim Carrey goes to a clinic to get his memory erased because it's so painful. So that's one thing that maybe one would think about what neuroengineering might do. Uh, Another possibility is this one is even older, although I think more people know about it, the Matrix, where a new engineer might construct a virtual reality world where the mind can tap into, serve different purposes, maybe to enhance cognition, maybe to serve our machine overlord. Finally, maybe the one that actually I thought of the first when I made it the slide, the movie Inception, where they're doing mind hacking, where they're trying to go into the mind of the people to change their mind for business espionage. So I think the movie has definitely some superlative idea on what neuroengineers can do in terms of changing the mind, but it gives you an idea on, where the imagination might go, right? In terms of where people think that these kind of technology can be useful. You can see that many of these are still, sort of a pipe dream, but the idea that this can be a very powerful methods to change the mind, I think, is definitely very pervasive in our society. But if you take a step back to, go back to reality and ask, what does a neuroengineer do? One could also even ask, what does even generally a biomedical engineer do? This is something that I think of more often, right? Because I work in the Department of Biomedical Engineering. A very clear and I think simple explanation for this is that a biomedical engineer combine biology. So this would be study of, for example, different organs. Here we have the lung, the heart, the brain on the top, stomach, intestine, and so forth. And for neuroengineer, obviously, we'll focus on the brain. And then there's also the engineering aspects of it, where we take engineering principle. A lot of these come from disciplines like electrical engineering, where we make circuits, prototype boards, and so forth, or mechanical engineering, where we're making devices. and then with the grand goal of taking both of these knowledge and tools to improve human health. So I think this is a fairly widely accepted and I think good definition about what a biomedical engineering do. And then in terms of new engineer, we're really focused on just one organ, which is the brain, but still with the overall goal of improving human health. So to give you some specific example, I think one very clear example now of new engineering that is, I would say, comprised of maybe like a third or even more of the kind of news and research that I see is the idea of a brain-machine interface. So here is one recent study where you have a human participant that lost her use of limbs, but she has a neural implant. So you can see the electrical array right here that's chronically implanted into her cortex, into the motor cortex, and then that allow the computer to read out the neural signal. There's some kind of decoding algorithm that allow that electrical signal from the brain to be interpreted, and then this allowed the user to drive a cursor on a monitor, which ultimately allow her to type. So this kind of technology is advancing quite rapidly. Here is just one example where we can control the cursor, but the most recent example now can combine with AI to directly read out speech, which is much faster and much more effective. But this is one, I think, clear example of neuroengineering where you're recording brain activity to improve clearly here human health in a person that no longer be able to move and in some cases communicate. There are also other examples of, I think, neuroengineering that we don't think about as much because they've been around for so long. One is cochlear implant, which is also a form of neurostimulation. Here, stimulating the inner ear to allow us to recover hearing for those who might be poor of hearing. And then one that has been around for longer is also deep brain stimulation, where electrode can be implanted into the brain and then powered by a pulse generator, much like a pacemaker for heart, in order to stimulate for conditions like Parkinson's disease or obsessive compulsion disorder. So these are other kinds of implantable devices that has been around for a long time that is already clinically approved and is actually used on individuals that are patient populations. A third example of neuroengineering I'll give you is devices. So here, there are a lot of advances now on miniaturizing machines. These are medical imaging devices that have been around in the hospital for a long time. Many of you might already have a MRI scan, but if you know MRI scanner, these machines tend to be really big and fill a whole room because they require a superconducting magnet. And there has been a lot of advances now to try to miniaturize the machine. So here I show you one example of a portable machine where the whole thing can be wheeled around. In a hospital, you can bring it. There's a lot of advantages of bringing it to different places, but you can also imagine these kind of machine would be also very powerful for deploying in underdeveloped countries where they may lack certain resources. But you can still, get a MRI scan, which would be very useful, for example, if you have any soft tissue damages. So I think these are examples of neuroengineering and what neuroengineers could do that are, I think, becoming more and more commonplace and important in the field. If you want to train to become a neuroengineer, I think just like where you come from in terms of needing the biology and also engineering training, typically the training path is either have a biology training or have engineering training, or ideally, be involved in both. For example, in the Department of Biomedical Engineering, like where I am, they ultimately give you both the biological understanding of the principle behind the tissue of interest that you're interested in this case, for example, the brain, along with training in the on the electrical circuits and the mechanical drawing that you will need to have that engineering mindset to solve the problem. So I gave some examples on what specific applications of what neuroengineering entail, but there are also a number of settings where this could, I think, be involved. And I think sometimes we limit to think about, oh, maybe like this involve working at certain companies, which is true. So I think a lot of it does entail medical devices. So for example, making physically these devices like deep brain stimulation, prosthetics, or software for them. But there's also a lot of opportunity for going to medical school and law school. I think having experiences in these areas now in terms of neuroprosthetics is also quite important for example, for things like patent law, where there are a lot of neuroethical issues. Is it ethical to put devices in people? What happens if these devices get obsolete? What are some of the patent and commercialization issues? That could also be a very good career. There are a lot of work also in the hospital and clinical setting because we make all these devices, the devices themselves and the imaging machine themselves also need to be routinely maintained and upgrade, and then just to make it work every day. So there are full-time engineers that work in hospital and in a clinical setting, for example, helping with ultrasound machine or computer tomography machine in the hospital. And then finally, there are a lot of academia and industry research going on. This is where I work because I work in a university, so in the academia. But not a lot of devices are currently approved. There's maybe a dozen devices. So there's a lot of active research both in the university setting as well as in company setting to come up with new devices. Well, which was, I think, also what make this field quite interesting because it's really a growing field where there's a lot of activity, unlike maybe other areas where you already have set type of solution. Here, I think there's an opportunity for growth and new technologies to come into play. So this is an idea just to illustrate that idea and how there's just a lot of excitement in this area, both in terms of research, but which reflect in terms of money. This is a plot showing you all the recent neurotech startup that's now coming into the space. So on the left is a list of just logos of various kinds of startup. Some of them might be more famous. Most of you probably know of Neuralink. That's the one that's led by Elon Musk. But there are many other ones that have come on board that are getting quite big. For example, Kernel, Synchron, some of these companies, as well as some of the other companies that are younger. And there's also established company in this area, for example, Medtronics and Boston Scientific, that have existing already businesses in these areas. On the right is a recent chart by a venture capital firm showing you just the investment in this area. It's definitely growing. I think there's a recognition that brain modulation technology, the way to change brain activity, could be very useful for just the growing mental health market, as well as the growing neurodegeneration market. There's also a lot of ideas on how these technology not only can be used for treating diseases, but could also potentially be used for enhancing cognition down the road, or maybe even as interfaces for computing. So yeah, I think that's near the end of the first part, but I just want to give you a sense also just academia, which is what I'm most familiar with, which is what one of my day looks like. You can also work in neurotech and work in academia. I interact a lot with the students in my lab, as I'll tell you about the research that we do. We talk a lot about the science. I sit in my office a lot, which is where I am right now. Actually, if I look out, this is the exact view. I'm sitting on there's a couch right here where I'm looking out the window, exactly this view. But I write a lot of grant proposals, think about the research and hold meetings. But we also work in the lab a lot to make instrumentation and do the actual lab research. And then as well as planning research. Here, Neil is a graduate student. And I'll talk more about, again, this process in the rest of the presentation. Okay, so again, this is the outline. Hopefully you get a sense on what neuroengineering is about. I gave you some examples of application and products. I gave you some field where I think there's a lot of activity going on. Hopefully some of them you might not think about. And then again, happy to talk more about these opportunities at the end of the presentation. But now I'll switch a gear to tell you a little about what my lab does and where I think that research fit into engineering, which is again a different aspect of it that you might not think about as part of neuroengineering. So very broadly speaking, my lab, we're very interested in how drugs work. Drugs are quite important because if you think about how we treat different kinds of diseases, even in complex diseases like mental illnesses and brain disorders, drugs are indispensable. If you think about the treatment options, about 80% of the people with mental illnesses, when you go into a clinic, the first line of treatment are drugs. I'm not saying that that's because drugs are the most effective, but mostly because there's also cost issue as well. But there's undeniable that drugs help a lot of people, and they're very commonplace with treating mental illnesses. So in my lab's research, we're interested in what drugs do in brain circuits, because ultimately that's how we think it leads to behavioral changes. And then some of our goal is that if we can understand what drugs do to brain activity and brain connections, we can maybe start predicting how new compounds might influence brain circuits and then be able to predict whether those might be good drugs or not. So in that way, we think our research is quite important for preclinical research before you even test a drug in clinical trials to evaluate whether those hundreds and thousands of chemicals that you created, whether those compounds could be useful to be tested even for drugs. The other thing that we have high hopes of is that if we can understand how some of these drugs work in the brain and why they might be therapeutic, maybe this allow us to do, for example, some of those neuromodulation that I showed you before, be able to tweak the brain activity maybe together with drugs or in the absence of drug as another way for enacting some of these therapeutic. So here, leveraging the reason for why some of the drugs work, but in service again for developing therapeutic. So those are the two goals and hopes of where I see this research brings. One of the drugs that my lab recently got quite interested in is a psychedelic called psilocybin. So psilocybin and the active metabolite of this drug, psilocin, can be found in the so-called magic mushroom that you might have heard of. There are about 200 species of these magic mushrooms. Here I show you a variety of species in the different locations in the world. Here there's a psilocybe liniforme that's come from the Pacific Northwest, including in Canada, in Vancouver, where I grew up. You have the psilocybe mexicana. This is in Mexico, where the compound initially get isolated. And on the right, you have a species that's found in Australia and New Zealand. So these mushrooms has been all around the world. Actually, people know for already a couple thousand years that these mushrooms, if you consume them, give you very profound behavioral effects. But it's only recently that been trying to test it for psychiatry purposes. In terms of the behavioral effect, you might know these as mostly psychedelic. So acutely, they give very perceptual and subjective effect. Just to give you an idea, here's one excerpt from a female participant that's taken one of these drugs. She wrote, "My inner vision became a universe filled with colorful waterfalls, glistening like stars not bound by gravity. They floated in the air and folded around each other, totally quiet. The streams are infinite and beautiful." And on the left, you see the picture that she drew about that experience. So you can see that these experiences, again, is highly perceptual. They causes very colorful imagery, although these experiences tend to be very short-lasting. So after oral intake of the drug, you might last for about 4 to 6 hours or so, and then by the next day, it will be completely gone. But the reason why these drugs are very of interest now for psychiatry, and you might have read it from a variety of newspaper and media outlet, is that they also seem to be quite useful for treating mental illnesses, particularly for depression. So that's been shown now in a number of phase 2 clinical trial in different places, like in the UK, Imperial College London, in Johns Hopkins in America, in Zurich in Europe, and then by company Compass Pathway as well as by Uzona Institute. So on the right here show you the main result from a clinical trial. This one is by Uzona Institute. You can see that they have about 50 people taking a placebo, niacin, which is this vitamin, and then 50 people receiving psilocybin with psychological support. What you can see is that there's a very substantial decrease in the depression symptom in these people with major depressive disorder. Baseline, and then 2 days, there's not a difference, but by 8 days, you see that difference beginning to emerge. And what's most striking is that 43 days after that single administration, you can still see that substantial decrease. So this is quite different than existing medication where you have drugs like Prozac or Zoloft that if you take it, you have to take it every single day, and it takes a while to work. Here you see that this drug has a relatively fast onset of about a week, and then most importantly, seemingly one dose can lead to very long-lasting suppression of these depression symptom, which, if been true in larger clinical trial, could be quite transformative for psychiatry. If you can imagine how one treatment could lead to some fairly long-lasting remission of the symptoms. So that's why there's a lot of excitement in psychedelic as used for depression treatment. You have these headlines, and I encourage you to read some of them to know more. But New York Times said the psychedelic revolution is coming. Psychiatry may never be the same. You have Newsweek saying that there's a new treatment for depression, and they have this cover with a bunch of magic mushrooms. But I've been also collecting headline. With a lot of these news, there's also a lot of hype. So also there's a lot of, I would say, irresponsible kind of headlines. Here you have a headline about how elite golfers are turning to mushroom and psychedelics to dominate the PGA Tour in golf. Here you have another headline from Rolling Stone about how people are trying magic mushroom for depression, but then accidentally meeting God. And then finally, this is my favorite, NPR, that says the National Park Service wants humans to stop licking this toad. So here they're referring to a toad called a Sonoran Desert toad, which has a psychedelic called a 5-MeO-DMT. So people have been seeking out this toad now in the national park in the BLM lands, except that the toad are actually now under a lot of pressure and may go extinct. So the Park Service want people to stop seeking them out. So what I want to tell you is that there's a lot of interest in doing research with these compounds because of their potentials for treating psychiatric disorders. And then next, I'll tell you about what my lab has been doing in terms of understanding what these drugs do in the brain. So we use mouse as a model, mostly because we can investigate quite deeply about what these drugs do in the brain. And the way that my lab approached this is from the angle of a neuroengineer. We use a lot of sophisticated technology and cutting-edge tools, primarily imaging tools, optical microscopy, to study what these drugs do. So on the left, you can see one of these microscopes that is in my lab. So you can see here, there's a lot of optics. The laser light comes in through this periscope. Here, there's a galvo resonance scanner to steer the beam at a high rate. And finally, it comes down on an objective on the left right here. This is a video of a mouse now that's on a fixation and is being imaged in the microscope. You can see the mouse is very comfortable. Sometimes it's drinking water, it's running on a ball. And then we're very interested in what happens in the brain before and after some of these drugs to get a sense and get an idea on what the drugs do. So this is some of the images that we can get from the microscope. If you come to the lab and check out the microscope, it's a kind of microscope to a two-photon microscope. So here we label the brain cells in the brain using fluorescent proteins, and we're imaging the top of this cell. So if you know neuroscience, this is a neuron, and then these are dendrite that comes out that receive the input. Here you can see that we're imaging those dendrite on the top. And what you can see is that not only you can see the dendritic branches that comes out, but you can see the little protrusions that come out of dendrite as well, which are the dendritic spines where the neurons would make connections. One of the work that's done by the lab postdoc Ling Xiao, she's very interested in how these drugs might change the neuronal connections, these dendritic spines in the brain. So in her experiment, she would do a surgery on the mouse, then perform these imaging twice for 2 days before the drug administration, give the mouse a single dose of psilocybin, which is a drug from the magic mushroom or saline vehicle, and then image it every 2 days subsequently, and then with one final imaging session about 1 month later. So these are the kind of images that we can get in the lab. You can see that here's a segment of the dendrite. And what Ling Xiao can do is to go back to the same area every single imaging session in order to track the stable dendritic spine, which would be the stable neuronal connections, as well as the new and eliminated spines to look at how the drug might influence the neuronal connectivity. So here what she found is that one single dose of this drug could actually increase the number of these dendritic spine, suggesting that this drug, psilocybin, has the ability to promote the formation of new neuronal connections in the brain. So what here, this plot shows you that initially there's no change, but a day after the drug, what Ling Chao see is about a 10% increase in the number density. So there's more of these dendritic spines in the brain. And quite interestingly, this increase actually is sustained. So even at day 34, she sees this 10% increase. So this drug, again, increases the connectivity between these neurons in the cortex in the brain. The other thing that she sees is not only that these drugs increases the number of the connection, they also seem to increase the size of them, which means that the connections are stronger as well. So here you can see day 1, there's an increase in the spot size, and then this increase declined a little bit over time, and then eventually by 1 month, they go back to baseline. But there is a substantial increase in the period over a few days immediately after the drug administration, suggesting that this drug, psilocybin, served to facilitate the formation of new synapses as well as make them stronger. We're also very interested in what are some of the reasons for why this happened. So here I showed you different kinds of imaging that my lab does. Again, we specialize in optical imaging. We're applying these engineering techniques to study drug action. So here we're not imaging now the structure of the neuron, but we're imaging the dynamic signals from the neuron. So here, instead of going into the brain, we're just staying at one focal plane and imaging it over time. So this is showing you the image over time. And what you see is that whenever a dendrite flashes, when it gets brighter, is when there's a calcium ion flowing into that dendrite. So you can see that these neuronal processes are quite dynamic and they have different calcium signaling over time. So what PhD student Neil Savalia found is that after a single dose of psilocybin, there's also an increase of calcium influx in these dendrites, which we think are ultimately quite important for how it can structurally remodel it. This calcium influx, the calcium serves as a signal that drives the subsequent remodeling. And as a final example for what we do in the lab, we not only do some of these live animal imaging, but we're also looking at other kinds of imaging technology. So here's a new technology where instead of imaging a live mouse, we actually look at brain tissue that we take out from the mouse, but we image the whole brain at once. So what we do is we take the brain tissue, perform chemistry to clear the whole brain to make it transparent. Then we use a method called light sheet microscopy that allow us to take multiple horizontal sections of the brain image in quick succession. So show you how that looked like, I'll show you this on the right. So this is now a clear brain where you can see the brain images at different horizontal plane very quickly. And so here is a whole mouse brain image sort of all at once within quick succession. And you can see different cell label in green and different cell label in red. And all of these images can be registered to the atlas, so we know exactly where these cells are with respect to the brain region in the mouse. So instead of looking at very detailed view before of neuronal connection, this kind of imaging allows us to take more of a bird's eye view of the entire brain of every single neuron, although at a lower resolution. So one thing that we've been doing is we've been very interested to apply this kind of method to machine learning. So what we do is image a whole brain after various kinds of drug treatment, 6 different drugs, take all these images and then ask whether the computer can, for example, predict what drug was administered to the mouse. It turns out that the machine is quite good at doing this, taking the brain images and predicting what drug was administered. So here, give you one application where on the y-axis are the true label, on the x-axis are the predicted label. You can see the machine predict mostly accurately with about on average 70% accuracy along the diagonal, meaning that it predicted the label agreeing with the true label. And then here we'll give it one sample where it was actually not in a training data set. this compound called 6,4-DT and ask the computer to predict and classify what drug this is most similar to. This is a use case that will predict what will be useful for drug discovery, where you might have a new compound, you don't know what it is. You can administer to an animal, take an image of the brain using this whole brain imaging, and then ask whether it's similar to any of the drug that's already characterized. So I think this will be quite powerful for preclinical drug development and drug discovery. So with that, yeah, I mean, that's a pretty quick overview, I think, of some of the research that we do in the lab. Again, it's a bit different flavor than what I showed you before. But to echo the point, we're very interested in here the organ is the brain. Specific biological question that we're interested in is what drugs do in the brain. And as engineers, we're bringing in these new tools that we are building and modifying in the lab in terms of optical microscopy, laser scanning microscopy that allow for these very detailed, fine investigational neuronal structure, as well as these whole brain imaging techniques, with the ultimate goal to improve health, and then in this case, really to find treatment that can improve mental health. So the lab work that I described involved a number of people. As part of the lab, I also mentor PhD students and then postdoc, and then the lab is funded through federal grants and private sources. We also work with pharmaceutical company that also contract us to do different kinds of research. As a final part of the talk, I'll talk a bit about how I got here. So just to give you an example on how my own career has been and how I ended up doing this kind of research, just to give you, a flavor on how that happened. So this is on the left here is my career trajectory, and I'll go through each of the stage. But I did high school in Canada, and then did undergrad, PhD, postdoc, and then now I'm a faculty. I'm now 45 years old, so I'm 25 years now removed from the start of my college. So in undergrad, I did an undergrad degree in Bachelor in Engineering Physics. This was not a very famous school, but it's a good school in Canada called SFU, Simon Fraser University, is in Canada. And then we did engineering physics. I cannot play media, but what I do is learn a lot of electrical engineering and also do a bit of physics. And if I could play this video, what it would show you is an air hockey game that we designed. So here we have 2 rows of sensors that's mounted on these wood plank that's painted black. This is an air hockey game that you might have played as a kid. And then here is a servo that moves this joint that is play the air hockey based on the sensing mechanism. So this is sort of way back when, I mean, this is a long time ago, where we designed a system that would play air hockey with you. So the servo would move the puck, returner, so it would return the puck when we play, so you can hit it towards the side and then it will return the puck. And all it does is just to illustrate the kind of training that I got as a bachelor student, which was a lot of software, a lot of electrical component design. We learned how to use all these dials, these servos, a little bit mechanical in terms of creating some of these components in order to make an integrated system. And that's sort of what I was trained in, and also a good amount of physics behind it. So actually, when I then went to, before I go in that, I also encourage you to do more in your undergrad if you're interested. You can see I did 5-year undergrad, including a lot of study abroad in Sweden, and then later also in Austria. So I encourage you to take this time to explore what you want to do. For my PhD, initially, I wanted to do more physics. So I was very interested in superconductivity. Here's one image of a superconductor. But sometimes things go in different ways. So I was constrained by funding problem, having to switch my research. So I ended up doing a lot of optics. And this is where my interest started in terms of applying these to a biomedical problem. So I was building microscopes, and many of these microscopes were useful actually for neuroscience applications and for imaging biomedical samples. So this took 6 years, where I got a PhD in applied physics at Cornell University. This is my thesis, where I now develop the microscope imaging neurons and neural circuits and brain tissues. This is one of the images that I took actually as part of my PhD. At this point, there's a lot of people now with a PhD opens up doors to go and work in industry, consulting, policy, patent law, some of the things that I mentioned to you. In fact, I would say many of the graduate students now in my lab, some of the recent ones have also gone to work in pharmaceutical industry. One of the postdoc, Luquan, just did that, as well as consulting. Clara, a PhD student, just went there. And then also other things like policy and patent law. But for myself, I decided to go further in academia. This is where I got very interested in neuroscience and taking some of those imaging methods. So I pivoted again, but for postdoc, I went to Berkeley, Cal Berkeley, did more neuroscience research for 4 years before finally starting my lab at Yale School of Medicine. I was still, very well much known for what I do in imaging, but at Yale, they have a long history of developing drugs, particularly this drug, ketamine. And this is sort of where I pivoted my research to use some of that imaging method that I've get very good at to apply to study drug action and try to understand drug action. And this turned out to be a very fruitful research direction and what I've been doing ever since. So now, as academia, you tend to move around a lot. You already can see. And I recently moved back to Cornell, where my lab is now in the Biomedical Engineering department here. And this is a photo of my building, my department, when the weather is nice, not in the winter, in the summer, just like now. So yeah, as a final slide, I talk a bit about my career. I'll give you some themes. Again, I don't want to give too much here because I think ultimately these things, you listen to the story, you get what you get out of it. There's a lot of survivor bias. What worked for me might not work for you. For certain, a lot of things don't work for you because it's already been 20 years ago. The job market has changed. As I mentioned already, maybe if I had gone into engineering when neuroengineering is an option, I might have taken that path. But when I was an engineer, that was not an option. Nobody in my department does neuroengineering. There is no such thing as of that as a degree. So I think things change quite often and change quite rapidly. But some of the things that serve me well is that I do see that the world can change a lot and your interests might also change. With neuroengineering as a prime example, I think one thing to note is you should know what you're good at. So in my case, I know that I'm very good at putting kind of gadgets together and then applying them to solve problems. Some of the things I'm not very good at, which might actually be a good thing why I didn't end up going to superconductivity research. I'm not very good at things that gets very, very theoretical or very deep in math. So even though I have a training in some of the mathematical background, I know that I'm not very good going very deep into that end. So that's also one reason I didn't go very deep into things like machine learning, for example. On the other hand, I also know that I'm also not very good being completely in the sort of the biology side. So I think where I am right now in terms of combining the technology with a biological application is a perfect place for where my skill set is good at. Another thing is just always be learning. I think this is something I see often where I think people who are interested in learning and they continue want to innovate themselves, I think tend to be better in terms of what you can achieve. And as I mentioned, the world is changing and the discipline are changing. Many of the things that when I was growing up in terms of jobs, right, don't exist anymore. For example, working for a newspaper, or very soon maybe working in things like real estate or truck driving. I think there are a lot of jobs that's changing, and then there are new jobs that are coming out. What I do right now in terms of applying these engineering approaches to study drug is also new. And I think 20 years ago does not exist. So I think that's something to keep in mind. What you want to do right now, like what I'm telling you that might be interesting 20 years later could be also quite different. And sometimes it's going to be very hard to tell what that might be. And finally, again, just I think be kind and pay it forward. I think it seems like the world is very big, but ultimately it's not that big and the community is small. So I've seen it over and over again now where people have helped me and I've helped people, and then I always come back, and it's a small world. So with that, yeah, that's all I have prepared for today. I didn't keep track of the time, but hopefully that's not too long and you're still engaged. But I'm happy to talk more about any of that, or what is it to be neuroengineer, my own career, as well as some of my lab research. So with that, I think I'll stop sharing and we can talk face to face.
Yeah, fantastic. Thank you so much, Dr. Kwan. It's so incredible to hear more about the work and the advancements you're making in your lab, and also some of your own career advice and experiences. Thank you, everyone, for joining us today, and thank you, Dr. Kwan, for your time. So we hope to see you next time. Bye, everyone.